All right, in this chapter, our goals are to just do an overview of the inputs and outputs of nutrition, how metabolism works as a whole, and how we measure metabolism, which is actually body temperature. And of course, I'm going to hit a little bit on how we regulate that body temperature. Like I mentioned in the intro video, this is a long, hard chapter just because chemistry is hard. Cut it into chunks. And the more you practice with cellular respiration, the easier it gets. I promise. I promise you can do this. So let's kind of start with just food. So we spent all last week talking about all the things we eat and how we move it through the tubes and all that sort of stuff. Now we're going to talk about what happens to that stuff. We're going to talk about how we actually break it apart for energy and the nutrients that we need to build new molecules. And I'll hit on a little bit of the storage capacity for carbohydrates, proteins, and lipids. But you do kind of have this lifestyle where you eat a lot at once and then you go through a period of fasting. So we kind of go through these swings of all or nothing, especially like in the American diet where we tend to kind of have, either three or more littler meals, but then spaced out with lots of time in between. I did touch on this in my intro, but one of the things I want you to really think about and seriously think about is how nutrition is just a very misunderstood science. And part of that is because the science is just bad. It's not the kind of experiments with evidence that we would normally rely on. I know that you guys all take nutrition, and I'm not trying to, make trouble. But I actually think that nutrition is really confusing for people. And despite the fact that we are in 2020, we are not getting healthier as a people. We are arguably getting less and less healthy. And I think part of it is the bias that is in the actual guidelines for nutrition and how it tends to really favor certain types of bodies, certain types of people, certain types of diet patterns, and that those patterns just are not fit for other people or most people, to be honest. So, anyway, I look forward to reading about your reflections and just be open minded. All right, some just vocabulary. We've got catabolism and anabolism. So, catabolism or breakdown reactions, we're going to basically catabolize all of the stuff that we've ingested and digested, and now they're going to be absorbed. And then we're going to use that energy from the breakdown to power anabolic reactions. Like anabolic reactions are building, biosynthesis, making things from smaller parts. Those two pieces, catabolism and anabolism, are always coupled in the human body. The coupling is ATP. So ATP is often referred to as like the energy currency molecule, and it's just going to take the energy from breakdown, transfer it to the pathway for anabolism, and we will need to kind of always keep those two things together so that we don't lose energy in the form of heat. So, metabolism is really a full summary of the body's total catabolic and anabolic reactions. So, a lot of people think of metabolism as like, I don't know, how easy it is to say skinny or something like that. It's really a much more technical definition in AMP. It's really looking at the total energy that your body is burning and then using to maintain homeostasis. For catabolism, I've always remembered that cats break things and that's why it's breakdown. We will see that all of those chemicals that you've ingested are going to have energy in the form of chemical energy being released. And we'll see four big steps of the catabolism process: glycolysis, which is really just the breakdown of glucose, the Krebs cycle, the electron transport chain, etc. There's going to be a lot of examples of breakdown, but that liberates chemical energy so that we can do anabolism. And for me, I've always remembered anabolic steroids or like build up reactions. But of course, you're going to want to build proteins, you're going to want to build DNA and new cells and all sorts of reasons you want to have new kind of large molecule molecules assembled from their smaller subunits. But do remember, catabolism is breakdown, anabolism is buildup, and it's ATP that kind of puts those two reactions together. Usually in a single cell, you're never going to be limited by a lack of ATP. There's going to be just a ton of it. Like a billion is a lot, right? Anyway, the ATP is just a molecule with three phosphates in a row, and because they're kind of all stuck together, they kind of want to fly apart, basically. So we can allow that terminal phosphate, that third phosphate molecule, to pop on and off, and that's what we're either going to use energy to glue it on or liberate energy by popping it off to power those reactions. I will remark that the efficiency of the human body is really remarkable. I sit around a lot with my laptop these days and it gets so hot and I'm just like, wow, that feels like such an inefficient waste of energy to just have that much heat like at the bottom of my computer. But listen, the human body is so by far more efficient at capturing that energy than any machine. All you have to do is just feel the heat of things like your car or your fridge or your dishwasher and appreciate the fact that even though 60% of our energy is released as heat, which is just excessive, uncapturable energy, the efficacy here is really, really, really good. Better than any technology we've ever been able to make. Anyway, here's sort of a molecular structure of ATP. You can see there's three kind of phosphates. Those are surrounded by oxygen. And that terminal phosphate can basically come on and off. And that's where we're going to couple the catabolism and the anabolism. All right, next, when we're talking about energy, we are usually referring to an electron. So electrons are like those kind of weirdly orbiting, negatively charged particles outside atoms. They have a lot of just energy to do stuff. Now, we don't want free electrons popping off in the human body. It's gonna have to be sort of combined with a hydrogen ion. And why that is that for the negative electron, if we couple it with a H plus, like a hydrogen ion basically, it stays electrically neutral. So when you hear the words oxidation and reduction in biology, this is maybe not true for chemistry or some physics or whatever, but biologically, because we're always very careful to keep the negativity and the positivity balanced, when you hear the words oxidation and reduction, yeah, we're talking about an electron with the energy, but a hydrogen ion is also there to just keep it all neutral, particularly when it's moving across a membrane, just like we've been talking about all year. So reduction is one of those confusing words because we often think of reduction as a loss, right? But reduction here is an addition of a negatively charged electron. So the reduction refers to the reduction of the charge because you're getting more negative, if that makes sense. It's also getting more energized. Oxidation is a loss. So remember those terms. They will definitely come up. And I just kind of want you to remember reduction is a gain of electrons / hydrogen ion. Oxidation is where we're actually plucking those electrons and hydrogen ions off. So there's going to be some key molecules that will take those electrons and hydrogens. That is NAD or NADH. Do you see how it can have a hydrogen kind of stuck on there? Or FAD and FADH2. Usually people don't refer to those by their names because obviously they are very long. They just refer to them by their sort of chemical abbreviations. Okay, let's talk about phosphorylation for a second. Phosphorylation is like a tongue twister too. So we kind of have these three options for where we see a phosphate involved. And there's one that I can eliminate right now. Photophosphorylation is photosynthesis, and we really only see that in plants and bacteria. It doesn't happen in the human body, but I thought I'd mention it because it is a type of phosphorylation. The first two we will see, and I'll discuss those. First is substrate level phosphorylation, and this happens sometimes. We just glue a phosphate on a molecule. This is actually pretty common in the cytosol, which is the cytoplasm, basically. Oxidative phosphorylation is much more important to our discussion this week. I'll mention a few things that we'll see that are phosphorylated on a substrate level. They just come into a cell and then they get a phosphate glued to them. But oxidative phosphorylation is oxidation, right? That's where we're going to remove electrons and hydrogens from a compound that we have digested, probably, ideally, unless we're sort of self-digesting during starvation, where you're just kind of taking your body's like molecules and proteins and stuff. But for the most part, you're going to be eating. You're going to be stripping that from like your ingestion of its electrons and hydrogens, and then it's going to pass through a series of reaction pathways so that we can actually make ATP from them. And as we go through this chapter, I'm going to break it down into carbohydrates, proteins, and lipids, like those three key nutrients, because they actually have slightly different pathways in order to get ATP generated from them. And sometimes we might not actually want to get ATP. We might want to like store it for later and then use it off the shelf when we need it. So let's start with carbohydrates. Carbohydrates are going to be the bulk of our discussion here. And when we talk about the most simple form of carbohydrate, we're talking about glucose. And glucose is very I would say predominant in our diet, and you almost preferentially are going to take that glucose and use it for ATP. Now, you could store it. And I would say that humans do not have a lot of storage room. So we're going to preferentially store really compact, highly like caloric nutrients over glycogen. So it's much easier, I guess, to store fat than it is like carbohydrate. But there is a small amount of glycogen in the liver and also in the skeletal muscles. So one of the things that might help you as you go through this week is just some word root vocabulary. So glyco means sugar, and genesis is the generation of. So when we're doing glycogenesis or glycogen, right? It's the synthesis of glycogen, which is the storage polymer of carbohydrate. And like I said, you have only a limited amount of storage space available. You could also use that glucose to make protein. I think a lot of people assume that carbs and proteins and lipids are just kind of like these three very different categories of molecules. But what I want you to visualize is how they easily move from carbohydrate to protein, or carbohydrate to fat, or fat to protein, or fat to carbohydrate. I mean, they really are more interchangeable than people realize. We just have to manipulate them. So the other two options for glucose are to use it to build amino acids and proteins, or to store it as fat. The molecule that we store lipids into is a triglyceride. So the first thing that happens when a cell gets glucose is it's going to have that glucose come across the cell membrane, and it is substrate level phosphorylated. So this is just a simple diagram. This is the GLUT transporter. The GLUT just stands for glucose transporter. As that glucose comes in, it is attached with a phosphate. And then what's really cool about that glucose with a phosphate on it is that it can't diffuse back out of the cell. It can accumulate above its concentration gradient, because you don't want to just lose the glucose, right? You like want to bring it into the cell and keep it. So by phosphorylating it, it now is going to remain inside the cell. It can't fit through the GLUT transporter protein anymore. It's our first example of phosphorylation. Okay, now let's talk about glucose catabolism. Glucose breakdown is also known as cellular respiration. And this is kind of where we're going to connect a few dots here. We need to connect the dots of, why does your body need oxygen in the first place? Like, why? Like, it's this elaborate system, right? To get the oxygen, transport the oxygen on hemoglobin, deliver it to the tissue that needs it. Now we're going to see a role for the oxygen. We will also see how your body produces CO2. So as you are breaking down glucose, we will see that as it's chopped into pieces, that glucose carbon becomes carbon dioxide. And we have a similarly elaborate system of transporting and expiring that CO2 into the atmosphere. So this is complicated, but by breaking it down into 4 steps, I think we will be okay. The first step is glycolysis. So lysis is breakdown, right? And glyco is sugar or carbohydrate. So we're going to break that glucose, and this can occur in the cytoplasm of a cell. Like, this is just happening all the time. The next steps actually have to be around the mitochondria. So the mitochondria is that organelle, like the powerhouse of the cell. You remember that? We will see that the mitochondria plays a role in the next 3 stages, which is converting that broken glucose into a molecule called acetyl-CoA. The CoA stands for coenzyme A, but nobody really calls it that. It's just like acetyl-CoA. The Krebs cycle, which your book really likes to call this the Krebs cycle, but I will caution you that the more modern, like cellular biologists, call this the citric acid cycle. So anyway, Krebs cycle, and then the electron transport chain, where we will see the other form of phosphorylation, oxidative phosphorylation, like we're actually getting kind of that stripping of energy and making ATP. Okay, remember how I said the little like text is really small in these images? Just look this figure up, I promise. This is actually a really good one, and you can kind of see a summary of these steps. So the first step here is glycolysis. And here is where we're going to take 1 molecule of glucose, which has 6 carbons. We're going to break it in half into 2 3-carbon pyruvate molecules. Then we're going to take that molecule of pyruvate and put it into the second reaction, which is a conversion reaction or a transformation reaction of pyruvate into that acetyl-CoA. Now, one of the interesting things about acetyl-CoA is it's only got 2 carbons. So as we take our 3-carbon pyruvate, right? Because we started with 6 carbons with glucose. So 6 carbons split into halves are, each of those halves of pyruvate are 3 carbons. So when we take pyruvate and we transform it into acetyl-CoA, which is only 2 carbons, we have to lose a carbon. Every time we lose a carbon molecule, it pops off as CO2. So there's our first expiration. Now our acetyl-CoA is going to enter the Krebs cycle. And the Krebs cycle is going to turn and transform that acetyl-CoA, basically chopping off carbon dioxides as they go. And then we will see that Krebs cycle will enter the last stage, which is the oxidative phosphorylation stage, which is where we actually get that removal of electrons and hydrogens, and those electrons and hydrogens from the oxidative phosphorylation in the electron transport chain step is going to get glued to oxygen. So your whole respiratory system that's delivered the oxygen to the mitochondria is going to get glued with electrons and hydrogens and then transform into water. Yes, that's right. The oxygen that you breathe is now water. All of that whole respiratory system services this mitochondria. It's actually the only organelle that requires oxygen, which is just a profound thought. If you want to pause the video and just like think about that for a second, it's so cool. I just love it. Anyway, let's proceed. Let's talk about glycolysis. So here we have at the start of this 1 molecule of glucose. Remember that 1 molecule of glucose is 6 carbons, and it is in a ring structure. So when you snip a ring, we're actually going to have to snip two bonds of that ring in order to liberate it into halves, right? That's actually going to require some energy. Every time you break those bonds to break the ring apart, it's going to cost you 1 ATP. So before we can start glycolysis, we actually need to input 2 ATP as a starting energy so that we can get that 6 carbon ring into halves. Now those halves are called pyruvate molecules. But I also want to caution you that some textbooks have a slightly different biochemical pathway, and it is sometimes referred to as pyruvic acid. It is the same thing for our purposes. If you see pyruvate, think pyruvic acid. It's just like in some illustrations that you run across, you might go, oh, what's that? Well, it's the same thing. So it's just half of a glucose. So here's the other thing that happens in glycolysis. Once those two halves of the glucose come apart into two pyruvates, it's actually at a lower energy state. And that is not super important to me, but I want you to know that it actually produces ATP when we chop the ring apart. So it takes 2 energy to snip the original bonds, but once it's all done, it actually produces 4 ATP, and we have reduced NAD into 2 molecules of NADH. Remember, NAD is an energy like it's a converter, like it goes back and forth. It's going to accept the electrons and the hydrogens. We've basically stripped those away from the bond breakage, and we've just glued them onto those 2 molecules of NADH. So if I was just to kind of run through this reaction, we start with 1 glucose. We take 2 ATP to start this reaction. We spend those ATP to split the glucose into 2 pyruvates. And then as those pyruvates form, we create 4 ATP and 2 NADHs. Now, I do think it's good to subtract the cost. So we made 4 at the end, but it cost us 2. So the net from glycolysis is just 2 ATP and 2 NADH. Now, this happens all the time, you guys. In the cytosol, this is constantly happening. Like, most people eat a lot of carbohydrates. So there has to be a point where the next stage is going to be an either or. And that actually depends on whether or not you've got oxygen or you don't. So if you have oxygen, and let's face it, cells use a lot of oxygen. There's going to be some oxygen around. We will see this enter the aerobic cellular respiration cycle in the mitochondria. If you don't, and you might need a few seconds to actually deliver oxygen, right? We have a different cycle that occurs. That cycle, in the lack of oxygen, is the Cori cycle. And it is where you take the pyruvate and you basically take the energy off of it and you produce lactic acid or lactate. And I would say lactic acid is probably the more familiar term to you because I hear it in athletics and stuff. You know what that is. It's that burn from anaerobic activity. So that can continue, except lactic acid can kind of build up and it kind of gets cytotoxic. But you can get a little bit of energy from it. I mean, not the most efficient energy, but for a short burst, an anaerobic thing might get you through 30 seconds. Most of the time, though, you're going to fall into this aerobic cellular respiration path. So let's just recap. Step 1, we got to split the glucose. Glycolysis, 1 6-carbon glucose gets chopped into 2 3-carbon pyruvates. That costs us 2 ATP, but we get, as a result, a net 2 ATP and 2 NADHs. Keep those NADHs in mind. We're going to use them later. What? This is another one of those really awful textbook images. But what you can see from this image is that there's one aerobic pathway. That's the one that's going to enter the next stage, which is conversion to acetyl-CoA, and then the Krebs cycle, and then the electron transport chain, versus the anaerobic pathway, where we're just going to basically take the energy off of that pyruvate and produce lactate or lactic acid, and then boom, done. We will be focusing on the aerobic pathway. So first, we've done our glycolysis. Now we have our pyruvate. It has to be converted to acetyl-CoA. Remember, this is happening in the mitochondria. So everything else now is mitochondria business. And what we want to do is form from one pyruvate, one molecule of acetyl-CoA, except acetyl-CoA is a 2-carbon molecule. So we have to chop one of those carbons off. As we chop that carbon, that's where our CO2 comes from. That leaves us with basically a 2-carbon molecule that gets glued to coenzyme A to form acetyl-CoA. So I know it seems like you should get some ATP or something from that, but you don't. What you actually get is 2 CO2 molecules, one from each of the pyruvates that we started with. Remember, we're doing this per glucose. And we get one NADH from each of the pyruvates being converted into acetyl-CoA. So I really like to do this as a grand total per glucose, but if it helps to go 1/2 at a time, definitely keep track of those. And watch these questions very carefully. It is most commonly going to be asking you per glucose, so you might have to double what happens from this step. All right, the third step is the Krebs cycle, or if you're cool, the citric acid cycle. This is also occurring in the mitochondria. And you guys, this is such a complicated set of reduction/oxidation reactions. You could literally study this pathway for the rest of your life and still learn things every day about it. There's things we don't understand about it. I would actually say there's just some magic about it, but it's really cool. Here's what we want to do though. We want to start with that acetyl-CoA. Remember, that is 2 carbons. And then at the very beginning, that acetyl-CoA is going to combine with oxaloacetate or oxaloacetic acid, depending on what book you're looking at. Your book likes oxaloacetate, but what I'll tell you is that the acetyl-CoA is 2 carbons. This new molecule is 4 carbons. When you add that, 4 plus 2, we're back up to 6 carbons, but it's not a glucose ring. It's a citrate or a citric acid molecule, which is why this pathway is often referred to as the citric acid cycle. So what we'll see next is a couple important events. We will form, for each citrate molecule, 1 ATP. That's cool. We got to keep track of that. We will also form 3 molecules of NADH and 1 of the other weird shuttle molecule, FADH2. We will also, for each turn of the Krebs cycle, for each acetyl-CoA that we started with, cut off each of those 2 carbons. So we will see 2 molecules of carbon dioxide form, and at the end, you remember we sort of glued it to that oxaloacetate molecule? That gets regenerated. So that just recycles itself. Basically what we've done is we've just chopped the acetyl-CoA off and liberated those carbons as CO2, but as we've done it, we produce 1 ATP, 3 NADHs, and 1 FADH2 per acetyl-CoA. Now, remember, I'm kind of a stickler for summarizing everything per glucose. So you would want to double this because you have 2 acetyl-CoAs, which means we're going to have as a grand total from the Krebs cycle 2 ATPs, 6 NADHs, 1 FADH2, 2 CO2s, and a partridge in a pear tree. Here is a very simplified version of the Krebs cycle. I used to draw this on my mirror when I was in school. I'm actually kind of obsessed with it. Super complicated. Do not overthink this. You do not need to study this. All I really need you to see is that it's like this cycle where we're going to basically transform that citric acid molecule back into oxaloacetate. And as we do it, we're going to produce all of those things. And here's a summary where we're at so far. Let's go back to glycolysis. We started with 1 glucose. We took a couple ATPs to get it rolling, but what we made were 2 molecules of pyruvate, and also we netted 2 ATP and 2 NADHs, right? We next converted those 2 pyruvate molecules into 2 molecules of acetyl-CoA. That gave us 2 CO2s, but also 2 NADHs. Now, those acetyl-CoAs entered the Krebs cycle. And for a grand total from the Krebs cycle, we got 2 ATPs, 6 NADHs, 2 FADH2s, and a total of 4 CO2s. Now, the CO2s aren't going to be energy, but I do think it's important to keep track of them. We kind of want to focus on the energy, though, because we want to add up the ATP. So here we've already made some ATP, which is great. We'll keep track of those. But let's just add up the other things so far. For the NADHs, we've got 2 from glycolysis, 2 from the pyruvate conversion to acetyl-CoA, and then 6 from the Krebs cycle. So we have a total of 10 NADHs and 2 FADH2s. Now, the next stage is the electron transport chain. And this is where we're going to see something happen to those NADHs and FADH2s. What we're going to do is take those molecules to this transport chain. We're going to strip the electrons and the hydrogens back off of them, and we're actually going to create a concentration gradient of hydrogen ions. We're going to pump those hydrogen ions into the space between the 2 mitochondrial membranes. Remember, the mitochondria is weird because it has 2 membranes. So in that space between the 2 membranes, all of the hydrogen ions are going to get accumulated in a high concentration gradient. And as they flow back into the inner compartment of the mitochondria, it will be forced to basically form ATP, and the hydrogen and the electrons has to go somewhere, right? As that ATP is being produced, the electrons and the hydrogens are going to get glued to oxygen, which is gonna form water. And I've totally balanced this reaction for you, so don't sweat it. But it's good to know that you can't just forget about the hydrogens and the electrons. We're going to use them to power a concentration gradient and then power ATP synthesis. And then as they flow in, we're just going to be here, stick it to oxygen. Oxygen likes it. And hey, bonus, we just produced water. So here's what the electron transport chain looks like. I will say there's some cool things about the electron transport chain. This is actually where your body uses copper, boron, cobalt, really, really weird metals, kind of micro vitamins and nutrients, are basically the core functional piece of these electron transport chain proteins. But anyway, this is sort of the inner mitochondrial membrane. NADH and the FADH2s have brought all of the electrons and hydrogens that it's carried with it from all of these catabolic reactions, and it splits them apart. Actually pulls the electrons away from the hydrogens, and it pumps the hydrogens out. Look, you can see it's a total concentration gradient, and it wants to flow back through that blue blob, which is our ATP synthesis protein. And the hydrogen flowing back down inside the cell is going to be coupled with the phosphorylation of ADP into ATP, so bonus, produced ATP. And then we're going to take the hydrogen ion and the electrons and glue it to oxygen to produce water. This is a very interesting model, wouldn't you say? Perhaps it needs its own name. Yes, it has its own name. It's called a chemiosmotic model. And how it works is basically similar to osmosis. I mean, usually osmosis refers to water, right? So it's not exactly like it. It's kind of like a chemical osmosis model. But it's basically just taking a concentration gradient of hydrogen ions, letting it flow back down its concentration gradient, and harnessing that energy to produce ATP. It's so rad. Anyway, where are we at? Let's just look at energy for a second. So energy comes in 2 ways. We got the ATP proper, Or we have those intermediaries, the NADH and the FADH2s, that we can actually turn into ATP in the electron transport chain. So let's do a quick recap. And I'm going to leave the CO2s out of it. So glycolysis, 2 ATP, boom, 2 NADH. Conversion of pyruvate to acetyl-CoA, 2 NADHs. Krebs cycle, we got a couple ATP, but 6 NADHs and 2 FADH2s. Now, my math tells me I've got 4 ATP, 10 NADH, and 2 FADH2s. Now, the electron transport chain is going to take those NADHs and FADH2s, and there's a pretty direct mathematical conversion for the FADH. For each molecule of FADH2, it produces 2 ATP. So I want you to keep track of ATP at this point. So it's like 2 FADHs, 2 FADH2s, I should say, times 2 equals 4 ATP. Now, the NADH is usually 3 ATPs per NADH, but there are some tissues that are less efficient. So we do kind of see an average range. Your book doesn't really say that, but I want you to know that some tissues are not quite as efficient at getting 3 ATPs. They can only get 2. So the 28 to 30 is an average score, and I think it's just more realistic. So if we kind of just add up all the ATP, that gives us a range of between 36 and 38, but it's definitely 38 in a perfect maximum world, which is a lot of ATP from one glucose, don't you think? I mean, I think that's pretty efficient. Pretty cool. All right, whew, God, we got through glucose catabolism. Let's do anabolism. Remember, anabolism is a buildup. We have a couple of options. We can store it as glycogen. Like I said, this is limited. We see a little in the liver and a little in the skeletal muscle. And this is really just your emergency sugar reserve, like before you really start to auto digest your important molecules, like your muscles, you will have a little bit of glycogen that you can just grab. This is especially important if you do go through swings of fasting. Okay, let's look at the next one. Glycogen lysis. Glycogenolysis is basically the breakdown of glycogen from the liver and the skeletal muscles, and it's actually kind of complicated. So, you will do this if you are basically hungry, which is where your glucagon hormone is going to come on, or you're stressed, which is like the adrenaline/epinephrine. Okay, let's look at this word for a second. Gluco, carbs or sugar, neo, which means new, and genesis. We're making new sugar, making new glucose basically. So if you're making glucose from proteins or fat, which is, technically a different type of molecule, it is gluconeogenesis. And you can certainly do this if you need to break down stores into that super easy to get ATP energy pathway. So when you're starving, fasting, if you're not eating any carbs, basically you're gonna do this process, and it's definitely stimulated by cortisol and glucagon. Remember, cortisol is stress and glucagon is where your just blood sugar is low, but you can certainly get it from proteins and fats. It's just gonna take a little bit longer, and it's just a little bit more of a complex pathway to get it into glucose than just to start from carbohydrates. I think that's why your body craves carbs so much, is it's just a very instant energy. All right, lipid metabolism. We're moving on. So remember that, first of all, lipids are not easy to dissolve in blood or body fluids. We actually have a transport problem. And this is going to be a couple different options based on the density. And I would think of fat as being not very dense, but protein being quite dense. So depending on the ratio of fat to protein, we can actually categorize different types of lipoprotein molecules. Remember, they're always combined just for transport into either the very lowest density, which is a kilomicron, or a high density, which is going to have a lot more protein. So I'll kind of go through each one of these really quickly. The kilomicrons are super, super, super low density, and this is basically like fresh fat from the GI tract. Remember, it's going to go through a lacteal, that lymph vessel inside the small intestine. There will be reasons to have chylomicrons, but for the most part, we need to kind of convert it into the liver more storable form. So the liver is going to be like, okay, I'll modify you, and you're going to become this, and you're going to become that, and it's not floating around for very long. Now, very low density lipoproteins are kind of a little bit more protein than a chylomicron, and you produce these as basically a basis for a lot of molecules in the body. So you do have reason to have these. You're going to need to have lipids for, all sorts of hormones and things, but there is kind of this next level of protein density. Next, we have the LDLs, the low density lipoproteins, and these kind of have a bad rap as being the kind of molecule that deposits cholesterol on the inside of your blood vessels. I will say that you need low density lipoproteins. You need cholesterol is like the basis for all the lipid soluble hormones. It's in the phospholipid membrane of the cell. There's a reason that you have it. You just kind of don't want too much of it so that it kind of, accumulates in places where it doesn't belong. The high density lipoproteins are mostly protein, and these are the ones that will actually remove the low density plaquing on the inside of blood vessels. So if you've ever had like your total blood lipid chemistry checked, they will look at your levels of usually LDLs and high DLs, and you kind of want more HDLs and less LDLs, but I would just caution you that this mechanism is not perfectly understood. The good and the bad designation kind of bothers me. It kind of gives it a word charge. There's a system for depositing and a system for removing those deposits from your blood vessels. Overall, you can use lipids to produce ATP. The nice thing about lipid is it's full of energy in a fairly small molecule. It's about twice the caloric value as a carbohydrate. But for the most part, it is your preferential storage molecule, which makes everybody so happy. And that storage is going to occur in the specialized adipose cells. Where is your adipose? You might want to know. Interestingly, it deposits in very asymmetric places. The majority is sub-Q. So the subcutaneous fat is under your skin. It's the most variable region. So depending on your body size, you would have a thinner or thicker layer of subcutaneous adipose. There's always a little bit of around the kidneys, and that's kind of our next topic in this class, but your kidneys are just under the ribs, around your back, so they can be a little bit susceptible to mechanical trauma. So a little bit of adipose gives them a cushion. The omentum, remember the greater, the lesser omentum, those are the two fattiest ones of all the visceral peritoneum. The genital region, some between muscles, and a little bit around organs, and kind of it pads the back of your eyeball cavity too. But just note that there's sort of a distribution of adipose, and the majority is, the really variable is going to be the subcutaneous. That's like really much of what you would perceive to be the amount of adipose in a person. All right, lipid catabolism, also known as lipolysis, breakdown of lipid. We will see that the triglyceride molecules, the storage form of a lipid, can be split into two sort of important components, a glycerol and a fatty acid. There's lots of hormones that are all going to sound like lipolysis hormones. Those will be like breakdown kind of hormones. The glycerol and the fatty acid, you should know, have slightly different pathways, but they can both eventually be converted into acetyl-CoA. And at that stage, you should know what happens. Acetyl-CoA enters the Krebs cycle, and then from there, it's going to have the NADH and the FADH2 enter the electron transport chain. Now, there can be other pathways, for sure, but once we get it to that acetyl-CoA stage, it's pretty straightforward. I will just mention a little bit about ketones. Ketogenesis is the generation of ketone bodies, and basically a ketone is where acetyl-CoA molecules get stuck together. So if there's a lot of acetyl-CoA, like an abundant amount, you will find that they kind of form like these little conglomerations basically, and that's a ketone body. It's a ketone is like a term in chemistry. It's a certain type of bond, and we don't really care about that. But ketone bodies can get converted back into acetyl-CoA and enter the Krebs cycle. So basically, proteins and lipids can enter this pathway at a slightly different step. We can kind of skip the glycolysis step and go straight into the acetyl-CoA. I hope that makes sense. And there are some tissues that prefer ketones to other types of carbohydrate metabolic pathways. And it's just there's more energy that you can get from a ketone body because it's like a cluster of acetyl-CoA than a single. Lipid anabolism. So if we want to make lipid, you should know you can make lipid storage from anything. You can make it from, well, fats, of course. You can store protein as fat, and you can store carbs as fat. And it just kind of depends on the pathway. But overall, the big picture here is that carbs, lipids, and proteins, which are, in excess, they will be stored as fat. Okay, here's kind of a cool fact about protein. Protein is not usually stored. It's usually like used. It's like usually in process. So proteins, of course, are enzymes, right? They're like reactive molecules facilitating chemical metabolic reactions all over the place. You want to kind of think of the subunit of a protein as being an amino acid. And usually when we have amino acids, we just use them to produce new proteins. We don't have like a protein amino acid storage sac or something. Those are kind of just used as needed. Catabolism is a little bit of a different story. Your cells are full of proteins. They're functioning, right? If you have to, you can break those down. It's not preferential to do that, at least for very long, but you can use those amino acids to make other amino acids and other proteins, or you can burn those up into ATP by creating a conversion of an amino acid into acetyl-CoA, and then, am I sounding like a broken record? The acetyl-CoA enters the Krebs cycle, and then you know what happens from there. It's also kind of interesting that different amino acids specifically help facilitate cellular respiration. And I want to show you this picture for a second. This is not in your book. So one of the key differences is instead of saying pyruvate, it's pyruvic acid. But like I said, you're going to run into that. After the pyruvic acid, it is something you're familiar with, which is acetyl-CoA. And then if you look at every box, it's a different amino acid. I guess I should say that yellow boxes are different amino acids. And the point is that there are 20 different amino acids. They can all be at different parts of the Krebs cycle, either an enzyme that facilitates a reaction, or they can enter the Krebs cycle as acetyl-CoA, which I think is really cool. All right, protein anabolism. Making new proteins, you got to do it. You should know that some amino acids are essential, which means you have to eat them. Some are non-essential, which means you can make them. And by making them, I mean you can convert one amino acid into another, but there are 9 that you absolutely need to ingest. You can't make them or convert one thing into another. It's important that you have all 20 so that you can, produce all your array of complicated proteins. Okay, let's just kind of back up a sec here. Now, metabolism is every one of those reactions combined. The anabolic, the catabolic, the breakdown, the buildup, all of that combined to metabolism. And we want to now regulate that in a range of homeostasis. So regulating metabolism is going to depend on your state. And there's 2 states. There's the state where you're digesting food. And do you remember how long that can take? I mean, it can take 7 to 9 hours, depending on how much protein and fat you've ingested. It might just be a couple of hours if you're going out for carbs and a drink of beer, right? So we need to really separate the energy that it takes for digestion. I mean, all of that, the blood flow, the transport, the detoxification of the liver. It is so metabolically expensive to absorb food that it's its own state. And then you go through the stage of not absorbing anything. So the post-absorptive state is basically where you're fasting. And I would actually argue that a lot of people never go into a post-absorptive state. They're eating very frequently. So if you just kind of think about the energy that takes, the human body, I will say this, is actually adapted to have a period of no digestion. It allows for things to kind of rest a little bit. And one of the things that I find really interesting about intermittent fasting is that it taps into a very basic physiological principle. You should think about it. I mean, it's not what I would consider a fad diet. I mean, it kind of sounds like a fad diet, but it actually is just allowing your body to kind of like recuperate and use that energy for other things, because it is such a massive like metabolic expenditure to get all of those nutrients like processed. Here's the theme for absorption. Store, 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 store. You are going to, of course, break down some glucose into ATP, go through the whole process. You're going to see some glycogen, you're going to see some protein storage, you're going to see some fat storage. Basically, everything here is going to be storage related. This is just the end of my list that you can read it. How is it regulated? Insulin. So remember from 231, insulin puts sugar into cells. And we know that that sugar can come from a whole variety of places: carbs, proteins, lipids, you name it. But like insulin puts storage into cells. The post-absorptive state is like, fasting basically. And here's the deal. If you're fasting, you still need energy to power all of your organs. So you have to be able to withdraw storage just as easily as you put things into storage. So there are some tissues, particularly the nervous system, that really needs constant supply of energy molecules. So you're going to pull things out in the post-absorptive state, and the theme should be withdraw, 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 right? This is where you're basically going to pull things out. Are there hormones? Oh, heck yeah. These are going to be basically insulin and glucagon. Remember, those are sort of antagonist, opposite hormones. But there's other things that are going to play a role. Stress, of course, adrenaline. But the two key players here are going to be insulin and glucagon. All right, how do we measure metabolism? It is body temperature. And, body temperature is actually really an interesting thing if you just stop and think about it. A lot of people take it for granted, but that's the amount of heat energy that is lost from the lack of coupling anabolic and catabolic reactions. That's pretty cool. To really get a good body temperature, you need a core body temperature, which is usually either an ingested probe into the inside of your body or a rectal temperature. People don't usually like to take their temperature there. So what we really measure is like superficial temperature or oral cavity temperature sometimes. And that can actually really vary depending on the time of day or gender or age. So I don't think those types of temperatures are a super good measure, but they can certainly indicate if your metabolic rate is raised or is artificially low. I will say, we also need to talk about your basal metabolic rate, which is the minimum amount of energy that your body needs to be functioning when it is in a post-absorptive state. You actually get pretty hot when you eat. Like, it's just part of that process. Like, there's just so many molecular reactions happening. How do you make heat? Exercise, movement, that's a good one. Since a lot of that is going to leak out as heat when you just don't couple a reaction, this is a great way to stay hot. Your hormones can certainly play a role. We saw examples of thyroid hormone disorders in 231 where hyperthyroidism, you have a high body temperature, hypo, you kind of have a low. There's other ones too, but like, this is kind of just reviewy. Your autonomic nervous system is going to play a role. It's interesting. Like there's things about your heat production that are just, I think, kind of tying into all the things we've been talking about. I will say the metabolic rate itself tends to decrease with age. That can really vary depending on, how much conditioning you have, just all sorts of things. But we do expect it to kind of decrease with age. But there's, so many complicating things. Your body temperature does have a set point. It's kind of like a thermostat regulated by the hypothalamus. So remember that hypothalamus is kind of playing a key role in the autonomic function of the body. There's going to be receptors measuring your basically body temperature all over the place. And the effectors are going to be primarily muscles, but you could maybe get goosebumps in your skin. Those don't really do much for producing heat, but your muscles, like, you could get up and move or shiver. That's definitely going to produce some heat. I thought I would mention brown adipose tissue. Brown adipose tissue, or BAT, is kind of special in babies. Most people grow out of their brown adipose tissue, as they get a little bit more developed. But what's really interesting, I think, about babies is that we kind of like chubby babies, like when they get those little chubby rolls in their elbow, and you're like, oh, you chubby cheeks. They have to basically have an emergency temperature ignition system, which is brown adipose tissue. Usually adipose is white. It's not super vascular. It doesn't have a ton of mitochondria. It's kind of whitish tissue, actually. But in babies, it's brown, and it's brown because it has a lot of blood vessels and it has quite a bit of mitochondria. And the brown adipose tissue doesn't do much about fat storage. It's not really for that. It's basically if they get cold and they can't do anything about it. Babies are nonverbal. I mean, they're noisy, but they're not communicative at that really very early stage in their life. I mean, they may cry, but you don't necessarily know what they're experiencing, right? So if they're cold, they can basically light up their brown adipose tissue and they can get warm really, 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 really fast. I think a lot of people don't recognize how extra clothes and hats and all the cute little things that you want to dress your baby. I know if I had a baby, I'd dress them as a vegetable like every day, and then maybe little costumes like dinosaurs and stuff. Anyway, they are often overheated, which can be a concern because they have like this built-in brown adipose tissue that just like lights up. It does one thing, and that is heats their little bodies up. It's so interesting. There's some interesting stuff happening with brown adipose tissue and different like medications for obesity, basically. Thought I would review hypothermia. Hypothermia is where your body temperature gets too low and things just slow down and it's really cold. The cold thermal stress is probably the more natural way that you could become hypothermic if you're like stranded outside on a, really cold, icy day or something, or you fall into a frozen pond. There can be diseases that do it if you're just not metabolizing things at the normal rate. A lot of anesthesia causes hypothermia. I always experience this at the dentist because I cracked my tooth from COVID stress last term and I had to go get it fixed. My dentist is pretty nice about it, but the injections of anesthesia always make me really cold, and sometimes I get really shivery, so I always like take a hoodie or something just because it's really a weird sensation to me. But that is actually super common when people take drugs as they get cold. Burns can do it, and definitely malnutrition. Fever is on the other end of this, and I will just mention fever is an abnormally high set point. It's not random. Your brain ups the temperature, and it can do that to basically mess with microbial growth. It can speed up the delivery of tissue repair things. Everything works faster when it's warmer in terms of chemistry. I mean, you don't want to overheat by any means, but a little bit of a fever can be a very beneficial thing. You just don't want it to be so high that your cellular structure is damaged. And if you guys are interested, there's this article. And this was not actually an article that was like generated from scientists. It was a bunch of moms, actually. These moms of kids who had severe autism. Basically, like, they all noticed that when their kid was sick and had a fever, that the ability to communicate with their kids improved. And so they did this really interesting study looking at basically if fever is one way that they can somehow like break through with these autistic kids. And what they found is it's not temperature, but it's actually the speed at which some of the chemicals are moving around the brain, and it can make signaling a little bit faster. And while it's certainly not like a cure for autism, it's a very, very interesting paper. And I think it's just kind of one clue about the mystery of that disease. But anyway, I'm in favor of low grade fevers. Obviously, high grade fevers are dangerous, but there's something to be said with how your brain knows what temperature to set it at. With, of course, whatever is your problem, you still need to like address that problem. But I just think that's really cool about those moms. Anyway, hang in there. I know there's a lot. I have one final slide, and that's food intake. There's definitely going to be hormones. There's definitely going to be the brain involved here. Hypothalamus is kind of always the center of everything autonomic, and that includes, eating, hunger, all that sort of stuff. Do recall that there's a 10-minute delay for the satisfaction or the satiety center. You just got to stop eating. Slow down a little bit, and then you'll catch up to yourself. If you keep going really fast, you can go over. But there's going to be hormones primarily that increase appetite, some that decrease appetite. There's going to be some physical feedback from the gastric and the intestinal phase that are happening in the digestive system. It's a pretty beautiful regulatory path. Ooh, I just hit an hour. All right. See you next week.